Lisa.
Yeah.
Yeah. It's pretty cool, the islands themselves, Santa Cruz and Anacapa, which are in sight right now. At night, there's only maybe a couple buildings. I don't even know if that. So there's really like, you see the glow of probably the coast, Santa Barbara and Ventura in the background.
Right.
And so they're kind of backlit,
Yeah.
so you just see the shadow of the silhouette of the island from our perspective being on
That's
the
cool.
south side of it. Yeah.
Yeah, that's neat. Yeah, it's a reminder you're not that far from a huge metropolis on shore.
Right.
Yeah, the Southern California Bight just basically goes from Santa Barbara to Ventura straight into Los Angeles. There's a lot of open space in between.
And do you see, I think from where you are at the footprint, well, when you're able to see through, like, um, my brain's not working great. That passage, Anacapa Passage between Anacapa and Santa Cruz,
Yeah.
you can catch glimpses sometimes of the oil rigs that are all lit up.
Yeah, we do see a couple of them out there.
I just had the same thought, whether it was the squid boats or the oil rigs.
Yeah, I think the oil rigs have those like orangish lights.
Tom, for the sampling, we have almost all of the boxes full of sponges. So the only box that does not have a sponge in it is starboard box F, which has the scoop in there. So you couldn't mix any more sponges anywhere else in the bio boxes except for that one. But if you wanted to slurp some sponges, we've only used slurps 1 and 2. But as far as corals, I think you could, it looks like you could put them in any of the boxes, but I'm not sure how large these sponges are. I didn't collect any of these on my watch. Right. So, and then we've taken 3 Niskins and 2 push cores.
Thanks, Taylor Ann. Thanks for monitoring the side chat and getting Tom up to speed.
Yeah, no problem.
On
thank
what
you.
he can do in the next watch, because that'll be the final watch for this.
Yeah.
And then
I think Acanthogorgia is on the list to be collected. And I know Lizzie said, well, we can get it later or on the next dive. But we sure are in it now, so I don't know if Tom will want to do that or not, maybe on the next watch.
Good point.
So that's a bank, that's a little bank off here to the left.
Yes,
that's sort of
I see that now.
Yeah, you can see the V on the opercle and the dark fin membrane.
And sort of a white band towards the caudal fin area on the body.
Yeah, those bands kind of can come and go depending on
right
where they're sitting, so they're not always real reliable. And yeah, you on this one coming up, you can see white, but it's not the same white spots as the all the Sebastomas are red, and then those white, and then they have those white spots as opposed to these sort of white spots that are showing up on that bank down below. Not to confuse you.
Is the next dive the last? Is the next dive the last one of the expedition, or are we gonna squeeze one more in?
I think the next dive after this is the last one, if I'm not mistaken, right, Lisa?
No, I think we're going to do one really short one.
Ah, okay.
Cool.
So there's this, we'll finish up this one. We'll do the rock bridge. And then after that, I think there's one more for 8 hours.
Copy that.
Where is that
Mm.
one?
Let's look. So it would be 1838. Let's see if she put it in there.
I didn't see it on the map.
I'm looking at our watch schedule. So let me call that up. So according to our, she might not have logged the dive plan yet, but we're finishing up footprint now. We'll start the rock, whatever one we just said, I can't remember that. The rock bridge one.
The rock bridge, yeah.
Yeah, we'll start at noon today, basically, through the 4 a.m. shift. So we'll be at the beginning and the end, our group, for the rock bridge. Then the ROV's on deck for the 4 a.m. to 8 a.m. and then we'll do the North Santa Cruz Shelf from 8 a.m. to 4 p.m. And then that's it.
That'd
So
be a
it's
shallow
just a
one, I
really
think.
short one.
North Santa Cruz Shelf, I think would be a shallow dive.
Yeah, I think it's very short and shallow.
Oh, so they are going to do the Santa Cruz Shelf.
Yes, but just for 8 hours.
Okay.
Like the Petrali Soul Dive, short.
That looks like an abalone shell in the lower right corner that we just went over. Wish I'd noticed it sooner.
I missed it. It was just the shell, or was it an animal in it?
I think there was an animal in it, but maybe not.
Okay.
I just caught it before it went off screen, so I didn't look at it. It have very long to look at it.
There's another bank rockfish right there next to the sponge,
Yeah.
the salad bowl
Yep.
sponge.
About 25 centimeters long, judging from the 25 centimeters long from the judging from the lasers. Another little tiny sea bastomas we're just about to go over. Bank off to the left, one off to the right, another Christmas tree coral.
There's definitely a lot to see here.
Yeah, I don't know which crab that was we just went over, or do you know your stars? I don't.
No.
We need to study stars.
Study stars. Anybody know the stars?
Another bank sort of nestled in there. They're very well camouflaged for this habitat.
Yeah, yeah. And a Christmas tree. We have certainly covered a diversity of habitats over this period of time, haven't we? With the different depths that we've covered and substrate type.
You mean on this dive or just in general over the entire
the
cruise?
whole cruise, yeah.
Yes,
the whole cruise.
for sure. Yeah, for sure.
And when you're
We could
going
span.
along in the mud for hours on end or quite a while, and then you come upon a rock that's just got all kinds of stuff on it, you realize how important structure is, and in particular rock, hard substrate.
Ooh, was that a lingcod that just sort of swam
Yeah, lingcod
by?
went through.
Mm-hmm. How much more on transects?
We have just about 40 meters left to go.
Okay.
Seen quite a bit of trash too on this transect. I think this is the fourth piece of trash that I've seen.
Sorry, could you say that again?
I
I couldn't
think
really
I'm
hear you
seeing.
so well, Taylor Ann.
Oh, sorry. There's been quite a bit of trash on this transect. I think this is like the fourth piece of trash that I've seen.
Yeah, we're
Old
not.
This
Budweiser.
moment brought to you by Budweiser.
Yes.
Yeah, it looks like a Budweiser can.
Yeah, probably.
Yeah.
Tom called it Sebasti's Budweiser eye.
What else have you seen, Taylor Ann? I haven't noticed a lot of trash. I've been missing it.
Yeah, I've
Right.
seen, I think 3 cans and then a glass bottle.
Oh
So
boy.
seems like a lot of drinking
Yeah.
done over here.
Oh, and looks like we're getting
Yeah.
into a bit more complex habitat here with
Yeah.
some boulders. So I'm not sure how we should do this because we're going to probably be going into the next watch for during this transect.
We're still trucking
Do
along. We'll probably finish up in the next minute or so.
okay, great.
Yeah. It
I'll
won't
tell you
take.
how long
There's
it'll
there's
take.
That's great.
there's
It'll
the
be 4
lingcod
minutes.
giving us a great view. Smile at us.
Nice.
Yeah. Nice, yeah.
Nice.
You can see with that big gaping mouth how much food they can get in there in one gulp. They're known as voracious predators of the rocky reef habitat. And look at all those basket stars. Ooh, is
Now this
that
looks like a speckled. This rockfish coming up looks like a speckled to me. They're very pretty.
chewling cod?
Be a
That's
big
a
meal for
speckled
that one.
rockfish.
Ooh, yes.
Yeah.
But
Ooh,
going
watch
after
out.
it though.
Oh
Probably
man.
could.
And he just came back, so must have missed. Yeah, that
There's
lingcod was going after him.
right, and there's
Here's
another
another
one.
lingcod. Yeah.
So fish have this mechanism of feeding called gape and suck, where their lower jaw basically unhinges and opens up, and it opens up really fast, and they can just sort of suck in the prey.
All right, we're going to sign
by
off here.
this
We're starting
action.
to trade off around here. So thank you for the great watch.
Thank you. We'll see you at noon.
Thank you so much.
Thank you. Yeah, thanks for your great guidance on the mosaic.
All right,
Yeah.
have a good
Couldn't have
day.
done it
Good night.
without you at all.
Yeah, we just have 15 meters. Yeah,
Right.
so we'll just
So-
come to a stop and I'll mark it here and I'll let them know and tell you and we'll hold.
Thank you. So Di, do you want to stay on and then you
Perfect.
can talk to Erica and Tom?
Sure.
Diana? Okay, then I'll get off so
Sure.
that they can get on. Tom
Okay.
and Erica? I'll put it in the side chat
He
that I'm
thinks-
getting off.
I think Tom says they're good to go. They've got the scoop from what they heard us say and what you provided with the mosaic, and then Taylor Ann has filled them in. So
Okay, great.
I will
Then we're
tune
good to go.
well.
We'll be back at noon,
And I want to
everybody.
thank you all so much for a great 12 to 4 shift.
Yep, and we'll be back again.
Thank you.
Sleep well. Bye.
And but first let me record the end of the transect time. So if you don't mind, Tom, unless we're far off, that should be ending soon. I'll write it down. I'll just listen. So I'll tune out now so you can get on.
Thanks, Di and Lisa. These corals are amazing.
Good morning, Erica.
Hello.
And good morning, 4 to 8 watch as you get on.
Morning, Tom.
Morning.
Data, this is Nav. Data, Nav. Yeah, I just want to make sure we mark this as the end of transect. Okay. Thank you. T12. Thank you.
Yeah, Kurt called that spot where ROVs go to die.
Yes, we will be avoiding that spot.
Hey Science, this is Marley on video. We're still finishing up our handoff, but I just wanted to check in and make sure you guys are there.
We are here.
Is it Tom and Erica?
Yes.
Yeah, we're here.
Good
Can you
morning.
hear us, Marley?
Yeah, you guys sound good. Awesome.
Okay.
Thank you. Just confirming, just doing a comms check. Thanks.
Roger that.
So I got very excited last night when we found 2 Acanthogorgia on our dive. And now look at us.
Yeah.
Yeah, there's just a few here.
Yeah.
What are they called again? Acanthogorgia?
Acanthogorgia. And when we're all settled, we actually are going to sample one.
Well, Tom, Meredith said she thinks her voucher is actually from Footprint, so she'd rather have it from somewhere else.
I don't know if we're going anyplace else, though. I mean, we're going close to Footprint on our next dive, but whether we'll see it on that dive, or if we dive North Santa Cruz, which will be more muddy, we might not see it. So it's a choice.
Yep, your call. I'm just, yeah, I got excited and was ready to sample last night, and she did not seem that
Well,
it
if you
was a
saw in the
priority.
right. But if you saw in the chat, the only thing we really have left to collect are corals and some slurps of sponges.
Roger that.
Because all the
Yeah.
boxes are filled with sponges. So
Right. So you've got to have a coral.
well, that's about all we can collect unless we want to find those doubloons.
Those would be nice.
Hey, Tom and Erica.
Good morning.
Hey, morning. Oh, I can hear my echo. I really don't like that. So just to go over the plan that I received, it looks like we're going to be continuing to about Z3 on this side of the slope still. And then our objective should be to get onto the other side of the slope, the northern slope, and mirror what we did, kind of going slowly backwards. And if there's an opportunity for a 2D or 3D habitat photo mosaic, that we should try to target the other side of where we did it on the southern side. Does that sound like what you
That
guys?
sounds. Yep.
Okay.
It sounds just like the plan that we were told, and that
Great.
sounds good to me
Nice.
as a plan.
And another
Meredith
thing is, I think, like RV lead and the RV team, because of the fishing gear encountered, we're going to try to just do small steps. Even with the shallow nature of where we are, it'll just be good to just be real slow with what we're doing. And we probably don't want to do any more transects is kind of the plan that we have, if that's okay.
How about a slow transect? Would that be okay or no?
Yeah, we've been advised per Bossman. We want slow moves and small steps, things like this. So especially as we go across ridges and things like this, it'll be more imperative.
Roger that. Yeah, I know when we had our call yesterday, I told Nicole that we basically need to avoid the shallowest area because that is filled with gear.
Roger. Yeah, we have a whole bunch of targets on here for where there's previous sightings and things like this. You can see them in high pack.
Yeah, so we'll have about 150 meters to transit over that shallow area to get to the other side.
Roger that.
Okay. All right, so we can kind of continue on.
Yeah, before we go, can we actually fire a Niskin right now? Because I don't think they've done one for a while, looking at our notes.
Yeah, sure thing.
Yeah, we've had 3 done, so might as well take one in the Acanthogorgia field.
And it's at the transect, so we can justify it.
Yep.
All right, Mary, looks like we have 3 of them open there.
Yep, we have 4, 5, and 6 open.
Roger that.
Data I have this as 127.
Yes.
Thank you.
You have to pull it left-handed. Come on.
Tom's back.
We missed you, Tom. We like you. All right.
Pop.
Popped. All right.
All right, I'm going to need that sample number again. 126.7. Sorry. 1, 2. Two, 7.
Thanks, Kim.
And the pretty cow cod has come out to visit. The lingcod.
Oh, yeah.
We saw quite a few cowcod last night.
Yeah.
4 to 8.
Yeah. Deeply indented dorsal spines. And
And large.
And large, yeah. The largeness is the most obvious.
We're really going to do a quick zoom on the cowcod.
Sure.
Looks like you got two little putties there.
Oh yeah, I didn't even see that camouflage, dude.
The lingcod and the cowcod. The two non-cods.
Is anyone else hearing an echo?
Yeah, I hear an echo.
Yes.
Okay.
It's yeah, the echo. It sounds like it might be coming from Erica. Can you check? Make sure you don't
Actually,
have.
I'm thinking it's me, but now there should be no echo. Is that correct?
Testing.
Well, it won't be me talking.
Busted.
Yeah, it sounds good now. Up.
Yeah, because the other day I found out when my mic is turned all the way up, the headphones bleed through and the mic hears it.
Yeah.
So I just realized that it was turned all the way up when you said that, so I had to turn it down.
I think that fixed it. Yeah, I don't hear
I
an
hope
echo
so.
for myself, so.
That fish looks to be breathing very heavily. The pumping water through it goes.
It's so pretty.
Well, it was, I think, right at the end of the last watch, if this was the lingcod, it was chasing another fish.
Oh.
So it may have just done a sprint, or it may have swallowed one, and now it's trying to breathe because it's stuck inside someplace.
Yeah, that's like when I exercise with Marley. All right, so Jess, I think our bearing is just going to be the same 100, and I can do, should I do less than 20 meters at a time? Is that?
We can start off with 20 meters, and then if it gets weird, then we'll slow.
All right, so let's continue on, unless there's incentive to explore in the tether right now.
No, we can just keep going.
Okay, let's go.
So you guys got to watch all the fishies go to sleep last night?
We actually did see one or two laying on their sides as if they had put themselves to bed.
Just such a difference looking at it now than when you see it in the day with all the fish around.
I wonder what they're dreaming about.
There.
What was that one, Tom? Off to the left.
The red one? Oh, the fish?
Darker. Yeah.
That was another lingcod?
Oh, okay. Just darker.
Yes.
Not.
And sorry, that fish that was breathing heavily, the darkly colored one, that was also a rockfish? Just it didn't have
no,
it.
that was a lingcod.
Oh, lingcod. Got it.
Yeah, there is actually a group of fish called the ling, and there's an actual group of fish called the cod, and it's neither a ling nor a cod.
Great.
Don't ask me why they put them together. And the cow cod's not a cod, it's a rockfish.
Right.
So.
Don't get it.
There's another cow cod on the rock.
Yeah, so Erica was saying they get really abundant at these depths and maybe shallower.
We have seen
Shallow,
quite a
you
few
might.
in the footprint.
Yeah, there's two here. Yeah, the footprint has lots of them. They used to be extremely rare and not very abundant anywhere, and now by protections they have become pretty abundant in certain areas.
Nice. Each calcot is a sign of success.
Yep.
It's always the big species that get it the hardest.
Yeah. More lingcod.
They had closed an entire area to all fishing, Tom? What were the rules of the calcot?
All bottom contact.
Well, there's
Lots
three
of
of
lingcod.
those, yeah.
I like these upside down sponges. I think they live that way because we've seen them all over, and it's only those kind that are flipped upside down. So either they survive when they get flipped upside down, or they also grow that way.
Jess, what have you been doing?
So when Abby was on last night, Tom, we talked about sampling some more of these plate sponges. We did get a sample of the salad bowl, which are the ones that are more bowl shaped rather than just flat shells.
Roger that.
And I know we only have a few sponge spaces left. But
yeah, well, we could break off a piece and slurp it, because I think we have some slurps left. And that's a vermilion rockfish. It'll be interesting to see what time the fish get up. If it's before dawn or they wait till the light.
Those squid have been tracking us for like 8 hours.
Maybe they're feeding in the lights up above.
Oh yeah. ROV Texas.
This reminds me of where we landed last night, or not, yesterday morning. Where we landed in the sponge field, but now we're in the coral field.
Yeah, that was pretty neat.
Yep. Footprint is an amazing place, and that's why it's protected.
So we are actually on Footprint Reef now, correct? I heard you and Erica saying that we were going there later. Was that a different part of it, or?
No, we're on it right now. We're over it, all on it. Basically, this whole bank is footprint. It just depends on where you call the beginning of it from the seafloor to the top. I mean, the top's obvious, but where you start calling it down on the bottom. When we get up into the 130 meter range, you actually would have ambient light down here, which is pretty cool, depending on what's going on in the water column.
The squid are definitely clued in.
So these yellowy corals are called acanthogorgia. And in certain areas in Southern California, you hit fields of them like this, and this is one of them.
Very appropriate for the Golden State.
Have you seen fields in other places, Tom?
Yeah, so there's some fields of this closer to shore over by Wainimi Canyon. So if we went east of here.
See a line there? Yeah, we got a line.
Glad you see it. I don't see it yet. Oh, is it coming off that rock?
Yeah.
Yeah.
All right,
Okay,
I stopped the ship.
I see it.
Roger.
And there's a ratfish down at the bottom. Wow, and now it changes to this encrusting sponge, it looks like. White everywhere.
Don't see anything that way.
Oh. The other side is what I don't.
It's like that's a fish or an anchor there.
Oh, that's an anchor, yeah. Good eye.
Riley, you want to push in there, please? Yeah, it looks like an anchor. Nice.
It's a giant fishing hook.
All right, Marley, come wide. Doesn't look like it's under tension though, so it must have been snapped.
Yeah.
At least you know where one end is.
Yeah, no, where's the other? Marley, push in there again, please. It's like that's terminated there.
Yeah.
Yep.
Great. Come on.
Good eye. Talk about me seeing the little fish. You're good at seeing lines.
Always curious where the second end of these things are.
Might be
That
pretty
is a
far.
question.
Snap. Wonder if there's a word for fishing gear phobia.
Mm-hmm. Cautious.
That's another. Well, that's another adaptive trait. Very adaptive. It would be highly evolved.
Well, in that case, our ROV pilot is highly evolved.
She is. We know this. No, just we were talking about the other phobias the other day.
Not really a phobia.
No, I know. But definitely a schema that you see and you respond to with alertness.
Yeah, Calcott is that other one on Boccaccio, Tom? The one in the
Yep.
center?
Has a little melanism on the side, that black spot it had. And there's some baby Christmas trees on the bottom. It's interesting how the black coral crabs are now on the Acanthogorgia just because they're so abundant. Lots of lings.
Pilots, would it be possible to get a zoom on that encrusting sponge?
Sure.
Bottom right?
Bottom right.
Or even on the top of that rock, it looks like the same stuff.
Sure, we can try.
Looks like
Can't quite see, but we can try zooming in and you can let me know. Go ahead, Marley.
Okay.
Is there anything in zoom here?
Yeah, right? It looks so similar to that Ophelia.
I think it's
Is it
debris now.
Lophelia?
It looks like it could be because there's Lophelia on the top of the rock.
Yep, more to the right.
A little more to the right, Roger.
Yeah, and that's Cash at the bottom.
Yeah.
Left, sorry.
Oh, there's a Lophelia.
And yeah, there's live Lophelia there. There's mixed in Lophelia right here.
So not
Dead and
encrusting
alive.
sponge.
No,
Stuff
I lied.
I guess we only have 6 meters left, that's fine.
Yeah, we're almost done. We haven't come.
You all good there, Science?
Great,
Yes,
thank you.
thank you.
Sure.
The red Paragorgia on the top.
Yeah, right at the lasers, yep.
So that wouldn't be a good target for a 3D pirouette.
I don't think there's enough alive for that.
Yeah.
But if we find something similar, I mean, look at it. All that white is just rubble from there.
Yeah.
Oh.
That's a lot of rubble.
Yeah.
And that's not really suitable substrate for anything. It's so unconsolidated.
Maybe some small things.
Yeah.
You know, some small, I don't want to say infotile, but in
Nothing
rubble things that could hide in there. Yeah. Anchoring probably not, but little things that could crawl in it and around it. Kind of be nice. A lot of little super tiny holes for small critters.
Definitely complex.
Definitely. The cow peeking out of the hole.
Yeah.
It never ceases to amaze me the holes that they try to squeeze themselves into.
Yep. This is a nice little feature. Or big feature. And just like that, the Acanthogorgia is gone.
I know, it's like, why here, not there?
Tell me when you solve that.
I always perceive things as such a result of recruitment, but I know that may not be the case. But
yeah, it looks like there's space here.
Yeah. And whether it's like they recruited to this area as well and did not do as well, or it was just this very patchy recruitment. Because zonation in these benthic organisms, I can't imagine as any other way, really. But oop.
Yeah, they're not moving around.
Yeah. That looked like a friendly chase down there. Hey, buddy. Yeah, so I'm thinking maybe another 20 or so, or maybe even up to 50 meters along this slope, and then would that be a good time to start heading to the other northern slope?
Yeah, sure. Whenever it looks like we can cross.
Yeah.
Well, that would be good. Can we zoom in on the coral next to the lasers?
Sure.
If that's possible.
It's
a little tiny thing. It's a white branchy.
Oh, wow. Okay. Yeah, I got you now. All right, Marley, go ahead. That's good there. Is it that one there?
It's two of them.
Ah, it's not.
Looks like a sponge.
It does look like a sponge now. That's why I wanted to look. It didn't look like a normal coral, so interesting.
Interesting. How interesting?
Interesting as in, that's interesting. I'm not sure. I mean, if we're okay, we could try to slurp it. It's pretty small.
Yep, sure, thanks. Forward there, Marley.
Remember, you wanted something hard.
Oh no, this is great. I'm digging this, guys.
And the slurp, Jess's favorite things.
So Meredith is calling it a sponge called Fakey Gorgia.
Fakey.
That's really good. Does Abby approve of that name?
This is way too early for Abby.
No.
So we get to name things what we want?
We do.
Gorgeous.
And that drives her bananas.
But she approved a
Oh,
salad
it's the
bowl
banana sponge.
Banana sponge.
We got to stay on the food theme, so.
And Jess, we're settled out. Is that enough? That's good for you.
Roger. Let's try again.
And Meredith said Abby isn't awake to object. So.
The best naming hour.
And for all of you out there in the world, Abby is our sponge expert, who basically tells us what things are, and she's worked a different shift tonight, so she's asleep right now. So we're just flying blind, kind of, but we have some knowledge of it.
All right, Ben, you wanna?
We have a whole list of things to collect, and this isn't on the list, but it's in the list of other.
List of other, Raj. See how that goes. Sorry?
We're going to slurp this.
We saw some different branching ones right as we were getting off shift last night, but they were pointy instead of rounded like these.
Yeah, these almost look like little tubes. But I like Fakegorgia, so. It's a little fakey.
That's what I put on the collection sheet, so.
That's what I put in my notes.
Now it's
all right,
real.
Meredith, you got to name it.
Does that mean she knows definitively it's not a Paragorgia? Is that a genus?
Yes, that is a genus, and no, it's not a Paragorgia. Once you get close, you can tell this doesn't have any polyps, so it's not a sponge.
Oh, I see.
I mean, it's not a coral.
Coral. Okay.
Yeah. We know what it's not. It's trying to find out what it is. It's not a fish. Put that down.
Speaking of, if there are any sponge taxonomists listening out there, we have lots of samples that need identifications. I know Abby's working towards it.
We'd love to hear from you.
But we have lost our expert this past year.
Yeah, it's very sad.
And yeah.
Ooh, flush. All right, Kim, I'm gonna flush you. Mary, what sample jar do we want here? 3, Raj. All right, that should be sufficient flushage. All right, Kim. If I fall off, I'll try pushing in back to the surface. Let me know if you want me to get you a little more stadial. Yeah, I think that might be back there. Oh, it's right where the bar is there. Yeah. Oh, yeah, there you go. All right, Mary, we're set up on 3 here.
All right.
All right, ready? Coming on. We got ourselves some sponge juice. All right, got full on there. Nice. Huh. Try hitting it against the rock. Or we can put this in the toolbox if you guys want it intact like this.
I don't think we have space in the toolbox.
Okay, we can hit it against a rock.
Unless you guys want to put it in with the scoop on starboard, but that might be too hard with how it is on the slurp.
Yeah, the slurp won't reach there.
Okay, fine then.
Yeah, if there's space intact is nice.
Depends on where you guys think. I think everything's filled up with sponges, so I was told the only thing left was the slurps.
Yeah, Kim, get it, yeah.
If there's just urchins in one box, though, we could put it in with those.
No, we have sponges and a sponge already in with those urchins.
Ah.
Oh, there goes that idea.
I suppose we could
Staurocalyptus sponge.
park it on the porch.
That's what happens
grab
when you've
it
been
and take it
diving for
around.
over 24 hours. Everything's full.
And I misjudge on how crumbly this was.
Maybe we can cut it in half.
Stow it, move it, and cut it.
Yeah, perhaps we can cut suction on the rock there and then pick it up.
Is it too small to grab and take around to F?
We can grab it too. We were trying to see if we can cut it into bits here. But would you rather we put it in F?
What do you think, Tom? Save the slurps for crumbly?
Yeah, it's 6 of one, half dozen of the other, so I will, whatever you think is best. It doesn't look like it's breaking up.
Yeah, we can,
Oop.
we might still be there actually. Let me look down on the brow here. Still there.
Oh yeah, nice. So the scoop does not have a biological sample with it?
No, not yet.
So we could set it on the rock and use the jaws to put in there?
Sounds like a plan.
Then I think we really are only left with slurps.
For sponges.
Yes, for sponges.
As long as we have room for the doubloons.
Oh, we're emptying bio boxes for those.
Abby up here. Where'd all the sponges go? I don't know. They're gone.
Here's 20 bucks.
Disappeared.
20 doubloons.
Yeah, here's 20 bucks. No, 20 bucks is better.
Yeah.
We'll keep 20 doubloons.
Nice job, Kim. Is this a better spot to do that?
It looks so perilous looking on channel 3, like hanging over the void.